Salutations, viewers across the world. This is Chico Squared, the Medicine Science Guru, in collaboration with Former Media Educational TV. In this episode, we're going to be discussing the Biology Paper 1, Zimbabwe Schools Examinations Council for June 2024. So this is going to be a 4-part series, whereby part 1, we're going to look at question 1 to 10, and the subsequent parts 11 to 20, 21 to 30, and 31 to 40, respectively. Right? So this is going to be a bilingual lecture series. I'm going to be using 2 languages as media of communication. I'm going to be using the King's language, the English language, and I'm also going to be using the vernacular Shona for ease of comprehension. So fasten your seatbelts, ladies and gentlemen, as we delve into the important nitty-gritties that we need to understand in as far as this paper is concerned. So let's look at the question 1. Which branch of biology uses biological processes, organisms, or systems to make products that improve the lives of people? Right, biology is the study of living organisms. Biology is a very broad subject. Remember branches akasiyana-siyana. So among the branches of biology, we have anatomy, physiology, we have biochemistry, biotechnology, ecology, etc., etc. But the question here requires us specifically to know the biological field which is responsible for improving people's lives by making products. Let's look at the questions. A, anatomy. Anatomy is not the correct answer, because anatomy, we are looking at the study of structure of organisms, okay? Then biotechnology. Yes, biotechnology is the process. No field the biology, not dilane. And among some of the most important biotechnological applications is the manufacture of insulin. Insulin, the hormone that is responsible for converting excess glucose to glycogen, thereby preventing a condition known as diabetes mellitus. So biotech is the answer, but let's look at other questions. Cytology, no, cytology is the study of cell structure and function, and then physiology, we're looking at the study of function. So 1B is the answer for this particular question. Let's look at question 2 now. If the flame jumps out too high when lighting a Bunsen burner, what are you supposed to do? So we are required to look for the answer that is most appropriate in these circumstances. So if we look at the answers that we have shout to alert the others. Turn off the gas tap. Yes. Let's look at the others. Close the air holes. Blow out the flame. But the answer which is most appropriate is to turn off the gas tap of the Bunsen burner to prevent or to avert impending calamities that might occur. Right, question 3. And the polysaccharides are a class of dash. All right, polysaccharides are a class of dash. Right, A, lipids. Quit. Ma polysaccharides in there ma carbohydrates and it. So carbohydrates, these are compounds that contain carbon, hydrogen, and oxygen. And so my compounds that contain carbon, hydrogen, and oxygen, the simplest unit of a carbohydrate is known as a monosaccharide. In my examples, monosaccharides, glucose and fructose. And then, if 2 monosaccharides join together, they form what is called a disaccharide. In my disaccharides, in my examples, sucrose. Like sucrose is a disaccharide which is formed between glucose and fructose. And then, right, the condensation, polymerization, and then the glycosidic bond. Okay. So my lipids are up, my lipids, my triglycerides, they are made up of what? They are made up of glycerol, nature, fatty acid molecules are 3. My proteins, my polymers are made up of my monomer units and my amino acids. And we've got about 20 naturally occurring amino acids that make up protein of living organisms, but we're not concerned about those things because this is an advanced level concept that we're going to discuss in future videos. So my carbohydrates no answer, but let's look at nucleic acids. Right, my nucleic acids, my hereditary molecules, and my molecules are heredity, and so we have DNA deoxyribonucleic acid. We also have RNA ribonucleic acid. So they are made up of what are known as nucleotides. And the basic structure of a nucleotide, the phosphate group 5-carbon sugar, and then organic nitrogenous base. It can either be adenine, guanine, cytosine, or thiamine. Right, chimwe chaunogona kushandisa kana dauza complementary base pairing and hydrogen bonding iri pa DNA molecule. Unogona kuti apples are tasty, cookies are good. Adenine ichibonda nethiamine, guanine ichibonda cytosine. Then RNA, uracil, etc. Of course, we're not going to be discussing more about the biochemistry of nucleic acids. Right, the answer that we have is the carbohydrates, the polysaccharides. My examples, starch, glycogen. These are examples of carbohydrates that fall under what are known as polysaccharides. Let's look at question 4. The diagram shows stages in an experiment to test a food for a nutrient. The test is for dash. Right. Nyari kuti kapana pande kudai so handere. Pama food tests, chiri chaunofana kuziva ndechekuti 1, you need to know the reagents that are used in a food test. Secondly, you need to know the color of the reagent. And then thirdly, you need to know the color change that the reagent undergoes kana that particular food you're to test is positive. And it. For example, starch test one iodine solution. Iodine solution is yellowish brown, and iodine solution turns into a blue-black color in the event yekuti starch is present. So, as you can see, we have a food sample in water, and then we have a dropper rhodonizera sodium hydroxide NaOH, and then in a copper sulfate solution, it also added after that. Okay, this is a test of protein, and my proteins are going to test using the Albastix test, or we can also use the Biuret test. This is a test of my proteins. All right. Glucose. No, glucose no test one Benedict's solution called clean sticks. And then starch test one iodine solution. Then my fats are not test one emulsion test, the transparency test. So under the emulsion test, we are taking water. We're taking ethanol, we put it in a sample, take a piece of bond paper, apply my fetsi papo, so unaona pane transparency, meaning to say you can actually see through. Right. So the answer that we have here is a test for protein. Question 5. A cell is the dash, and then a cell is the dash. Right. Let's look at the questions that we have. Smallest part of an organism, basic functional unit of life, site of biochemical reactions, active part of an organism. My questions are biology, I don't papara like a crazy chicken. Especially multiple choice, because my answers are ngaripo. Akafana S and S are correct. Saka, you don't have to rush. You analyze through all the questions, bichana, up until you get to the most appropriate answer. All right? So a cell is a basic functional unit of life. Though the most appropriate answer, yoyo. Smallest part of an organism, that's a lie. Because smallest part, the cell in the smallest part, mukati cells, organelles are even smaller than the cell itself. Kuna na nucleus, cytoplasm, mitochondria, ana Golgi apparatus, vana endoplasmic reticulum, ribosome, lysosomes, all those things are smaller than the cell itself. So we can't comfortably say the cell is the smallest part of an organism. That's not true. That's not true. So the basic functional units of life, okay? Then the site of biochemical reactions. Mama cells, biochemical reactions. Hope. That's not the most appropriate answer because my biochemical reactions talking to an achitikira somewhere apart from cells. Okay? So the most appropriate answer is B, which is the basic function, right? The hereditary material. Right? Kati boy see ever side. It correct, but can analyze it. It is a site of respiration. No. The site of aerobic respiration in plants is known as the mitochondria. Right? The respiration in the phases, glycolysis, because it runs the Krebs cycle, because it runs the electron transport chain, right? Glycolysis in the cytoplasm, and then the tricarboxylic acid cycle, the Krebs cycle happens in the mitochondria, right? Our respiration is mitochondria, right? It contains cell shape, no. Cell sepu inowanikwa mu vacuole and vacuole ndo site ya cell inenge ine ma salts ma sugars ndo iri kucontrola futi entry and exit of substances by osmosis, because concentration ye cell sep, concentration ye vacuole ndo inodetermine what's going to enter, what's going to exit due to differences in concentration gradients anenge aripo ipapo. Site of photosynthesis, no. Photosynthesis happens in the chloroplast. So it contains the DNA, the hereditary material, deoxyribonucleic acid, the double helical structure of polynucleotide strands. So 6A is the most appropriate answer in as far as question 6 is concerned. Let's look at question 7 now. The diagram shows a specialized cell. The cell is specialized for dash. Before you can know what the cell specializes in, you have to first of all identify what the cell is, and all right. As far as my eyes can see, cell rubber by it's called an erythrocyte, called a red blood cell, and okay. A red blood cell is responsible for transporting oxygen. So if we look at question 7, C ndo iri kuratidza kuti ine chekuita netransportation of oxygen mukati. However, ngatibotarisa dzimwe dziripo. A, producing antibodies. No, antibodies are produced by my cells and my lymphocytes. My lymphocytes, they fall under my white blood cells. And remember, white blood cells are responsible for maintaining the sovereignty and the territorial integrity of the body, meaning to say to fight against my pathogens, to fight against my disease-causing organisms, my bacteria, my fungi, my actinomycete, my protozoa, etc., etc. All right? So a lymphocyte, they don't produce my antibodies. All right? Ingesting my pathogens in it one of specialized white blood cells are my phagocytes. So my white blood cells, they are divided into phagocytes and lymphocytes. Phagocytes are responsible for phagocytosis. That's the process of actually ingesting and digesting the bacteria to make sure they are totally eradicated so that the human body doesn't suffer the consequences of these immoral pathogens, okay? All right? So, transporting oxygen, transmitting impulses. Transmission of electrical impulses is done by my cells, are known as my neurons, called my nerve cells. So we have what are known as sensory neurons, we have what are known as motor neuron, and we have what is known as a relay neuron, okay? We're going to have a whole video whereby we're going to be explaining the nervous system, the function of these neurons, etc., etc., okay? So the answer we have here, number 7, is an erythrocyte. It's a red blood cell, and it's responsible for transporting oxygen. Question 8 reads, a detergent which contains enzymes will work best in water 8. Right? Panapa nyaya iripo ndeyekuti varikutaura havo nezvedetergent etc., etc., but the issue iripo apa ndeye-enzymes, enzymology, handeni, okay? Characteristics of enzymes ndo zviri kubvunzwa apa, handeni. Tine ma-characteristics hobho ema-enzymes. Enzymes are specific to the substrate that they catalyze. Enzymes are three-dimensional globular proteins that catalyze metabolic reactions in living organisms. Enzymes are sensitive to temperature. Enzymes are sensitive to pH. Right. So, panapa, varikubvunza nyaya ye-temperature ne-enzyme activity. So they've disguised question ye-temperature and enzyme activity vachitaura nyaya dzema-detergents. So you have to relate that concept to the, depending with the type of enzyme we are dealing with. If temperature is excess, the enzymes undergo what is called denaturation. They are denatured. When enzymes are denatured, it means the active site, which is complementary to the substrate, is destroyed. And if the active site of an enzyme is destroyed, it means that there is no complementary, the substrate is no longer complementary to the active site. Hence, the enzyme is done and dusted. Enzyme all right. So we can't talk of any temperature. Then cold temperature. Right. If the temperature is excessively cold, okay, enzymes are inactivated. Okay. When enzymes are inactivated due to cold temperature, when temperature is restored gradually, the enzyme activity will start to be restored as well. Unlike denaturation, When an enzyme has been denatured, there's nothing that can be done to reverse that. Cold temperature heat, because to tell at cold temperature my enzymes were inactivated, so the detergent will no longer be as effective as it is supposed to be. You understand? Right? Then what temperature and that enzyme in denaturation. So I heat a warm temperature. Warm temperature is the most appropriate answer to this particular problem. So 8D is the answer that we have. Let's look at question 9. We have a graph. Again, it's for enzyme activity. The graph shows the effect of substrate concentration on the rate of an enzyme controlled reaction. At point X, the active sites are dash. Right. Graph here show substrate concentration versus rate of reaction. And as you get substrate, the reaction is no longer increasing its rate. The reaction is now constant. So okay, what's really transpiring in as far as this graph is concerned, and how can we connect enzyme activity to what is actually transpiring on this graph? Right, substrate concentration and the rate of reaction up to a point whereby continued increase of substrates is no longer increasing the enzyme activity. Right. So at point X, my active sites and A unoccupied. That's a lie. Because you can always run my substrate because not my active sites A say are saturated. And it. Alright? So B no answer. The substrate, the active sites are saturated. But starting a certain number of enzymes and a certain number of substrates. And Sakatawanza some substrates to a point where all the enzymes are now occupying substrates, and increasing the substrate will no longer increase the rate of a reaction. So the answer at my active sites, they are saturated. Papan's destroyed, denatured, dana. Active site, apana anything should rather destroy, apana anything should rather denatured. Remember, in our previous question, Tatara Suskuti. Chineta guarantee enzyme denaturation in excessive temperature. And there's nothing that has been mentioned about temperature, and as far as this question is concerned, it's only about substrate and rate of reaction. So thick. All right? But remember, one characteristic of enzymes is that they are specific to the substrates that they catalyze. So one property that makes enzymes to be specific. Let's look at the questions that we have. Part A of the question, sensitive to pH. No. pH sensitivity has nothing to do with enzyme specificity. That B possesses active sites affected by temperature, protein in nature. Right. Nyama active sites no answer. Because the active site here enzyme in a shape yakafanana ne substrate. Test out the enzymes are complementary to the substrate. You know, the lock and key hypothesis. Lock and key hypothesis, yema enzymes, not about the enzyme and the substrate are complementary, just like a lock is complementary to a key. Kurati key no kinura pa door iri. Ndiyo. Ukazotora key no kinura mwe door. Aichavume because key iri complementary kwe certain lock ya irufana pinda ipapo. Okay? Ukatola salivary amylase that we digest ama lipids. How digest? Why? Because the structure of lipids yakasiyana ne structure active site amylase. All right, you understand what I'm saying? So enzyme specificity. Enzymes are specific to the substrate that they catalyze. This is only because of the active sites which are complementary to the enzymes. Viewers across the world, this was only the first part to the June 2024 Ordinary Level Zimbabwe Schools Examinations Council Biology Paper 1. We are going to be having part 2, part 3, and part 4. And we are also going to be doing the theory paper and also the practical paper. We are also going to be doing IGCSE, Cambridge, AS, and advanced level biology papers in our future videos. So to get notifications of what we are going to be doing in the future, be sure to like, subscribe, comment, and share with as many people as possible so that we enlighten the whole world, we enlighten the whole universe with the information that we are disseminating at this point in time. Ladies and gentlemen, comrades and friends, viewers across the world, this is Chico Squared, your host, the Maths and Science Guru, in collaboration with Farmer Media Educational TV. Be blessed and stay blessed, and I wish you all the best in your academic endeavors. I salute you. Chakwenda Chibaba.